Hello, everybody. I'm going to introduce you all to the first cell in our book, a remarkable cell called the Purkinje cell. When physiologist and pathologist Jan Evangelista Purkinje first drew this cell in 1832, the only techniques available at the time to enhance the visibility of the cell meant that to him, the cell looked a lot like the beginning of my drawing of the Purkinje cell, with only the soma, or cell body, and the trunk and larger branches of the dendritic tree visible enough to draw. It wouldn't be until 1875, almost 50 years later, that pathologist Camille Golgi would develop a technique inspired by photography development using potassium bichromate and silver nitrate to stain the cell black. This process is now known as the Golgi stain, or the method of silhouettes. At this time, armed and inspired by a new technique for visualizing the Purkinje cell, neuroanatomist Santiago Ramon y Cajal would also begin developing his theory about the form and function of such a beautiful and mysterious cell. Despite their opposing ideas about the form and function of the cell, and even whether it was a cell at all, Golgi and Ramon y Cajal would eventually share a Nobel Prize for their collaborative contributions to neuroscience. Firstly, Purkinje cells are mostly concentrated in the cerebellum above the spinal cord. Like most neurons, they are made up of a soma, or cell body, where the nucleus lives, dendrites, where they receive input, and an axon that transmits signals as output. But what makes these cells unique is their flat configuration and the tree-like shape and density of their dendrites. The reason they are formed this way has to do with their function in the cerebellum. For instance, these intricate cells have evolved to quickly process large amounts of sensory and motor information, forming the foundation for skills like muscle memory, hand-eye coordination, balance, and even reading. In other words, they make dancers more graceful, athletes more precise, and readers more efficient. Without the immense synaptic density of their dendrites, Purkinje cells would not be able to process as much information as fast as they do, which is necessary to quickly send the output signals necessary for real-time adjustments and fine-tuning. Additionally, Purkinje cells also have highly dynamic, weak synapses, which allow for more synaptic plasticity so they can learn from mistakes, which is another factor in their ability to adjust in real time, but also what helps them refine movements for muscle memory. Their extreme efficiency of information processing also gives rise to their flat configuration and their unique orientation in the brain. Purkinje cells are oriented parallel to each other, densely packed into a single row, forming the Purkinje layer. An interesting fact about Purkinje cells that I've learned outside of the course materials is from a study by Hadi Vasiliu, Grunwald, and Davies Jones on how the body's autoimmune response to gluten can contribute to neurological damage. Their findings suggest that IgG anti-gliadin antibodies can mistakenly bind to proteins on the Purkinje cells, which cause them to atrophy. This condition is called gluten ataxia. I hope you have all enjoyed this introduction to the form and function of Purkinje cells. This presentation was created using a time lapse of myself drawing a Purkinje cell, which took about an hour and a half to complete due to the sheer density of its dendrites. Here's the final look and my references. Thank you and goodbye.